kingdom fungi then, we're going to look at, particularly going to look at Rhizopus, which is the one in the diagram, but just a little recap on the kingdom fungi. They got a kingdom all to themselves because they don't fit in with any of the other kingdoms. They're not plants, they don't photosynthesize. Just a few little notes on them over here. They reproduce by spores. We'll see that with Rhizopus. That's the picture diagram there. We'll look at that in a second. They're eukaryotic, so they don't fit in with the bacteria, which are prokaryotic. And like I said, they don't fit in with the plants because there's no chlorophyll. They don't photosynthesize. They do have a cell wall. Their cells have a cell wall, but it's made of not cellulose like plant cells. It's a polysaccharide called chitin. So the cell walls of fungi are made of this stuff, this chitin. They are mostly saprophytic, mostly saprophytes. In other words, they grow on dead organic material. Mostly saprophytes. Some are parasitic. Some will parasitize things like insects, and if the spores land inside an insect or on an insect, then they can literally grow inside it and grow from the inside out and kill the insect as it grows. And then some of them a lot of them live in soil. A lot of them are decomposers. They're extremely important as decomposers. Like I said, the saprophytes, a lot of them are really important in the recycling of nutrients as decomposers. Some of them, like I said, are some of these soil dwellers will be able to, they catch nematodes, little tiny worms that live in the soil. They're able to they make these kind of lassos out of the threads that their bodies are made of. That these fungi made of these things called threads, these things called hyphae. These are what the sort of the units cells, if you like. Although they're funny that the cells aren't, that the nuclei aren't separated in these long filaments like you see in the picture. But some of these hyphae in some of these fungi, they form kind of lassos, and they in the soil. Then when a worm, tiny microscopic worms, move through these lassos, then they kind of touch these little hairs, these little sensors, and the lasso tightens really, really quickly and traps the worm, and then the fungus grows around it and digests it. Digestion then with fungi is extracellular, so they don't engulf things like the amoeba does. They just produce enzymes, and the enzymes then digest the food outside. Substrate is outside the body of the fungus, and the enzymes are chucked out onto the substrate, onto the food, and then the digested products of the enzyme action are absorbed into the fungus through the cell wall into the cells. So hyphae, by the way, a massive hyphae or a bunch of hyphae collectively is known as mycelium. Mycelium. So hyphae, one hypha, two hyphae, they're the threads. Mycelium is what you call a bunch or a mat of hyphae, and that's the. We tend to think of mushrooms or fungi in general as mushrooms is the first thing that comes to mind. Mushrooms are only the fruiting bodies. They're only the thing you see above the ground. Underneath the ground is really where the mushroom is, where the fungus is. It exists as these hyphae generally, and a large area or a bunch of hyphae is called a mycelium. Okay, so we'll just look at Rhizopus in particular. This is Rhizopus over here. Rhizopus. And Rhizopus is commonly known as bread mold. Have you ever seen a piece of old bread, or bread that's left too long in a bread box or something in the bag? It can sometimes form this black mold. Classically, it's a black mold, and that's what Rhizopus is. When you see it as a kind of black threads, and also with black dots. You see black dots forming if it's left long enough. What we're looking at in this picture, the black dots, in fact, are these circular things that you see, and there we'll look at what they are. They're full of spores, and they're the fruiting structures. They're the reproductive structures. This diagram here represents what we would call the asexual reproductive structures. Asexual Rhizopus, like lots of fungi, they generally will reproduce asexually, and only will switch to a sexually reproductive method when things are tough, when the conditions are adverse. So when things are getting difficult, or the conditions are getting unsuitable for the fungus, then they'll produce a special kind of spore we'll see later on by through sexual reproduction. But let's look at how the asexual reproduction, this is the everyday sort of reproduction that Rhizopus and lots of other fungi will go through. First of all, this word hyphae again, all these threads that you see, all these lines are all hyphae, they're just different, have different functions. So these hyphae here on the bottom, I'll just these hyphae here are called rhizoids. So just to label these, sorry, it's a bit too thick. So these are called rhizoids, these labels here. I'll just spell that for you. So rhizoids, R-H-I-Z-O-I-D-S, that's rhizoids. These are for anchoring in the substrate. So this will be underneath the thing that the mold is growing on. So bread, in the case of bread mold. So these are for anchoring. They also will produce enzymes which will for digestion, which will go into the food and then act on the food. I guess if it's bread, then you're talking amylases will break down the food into sugars, and then these rhizoids will absorb the end products of digestion. But another kind of specialized hypha then would be these ones here. These are called stolons. This is a stolon. So the stolon is S-T-O-L-O-N. They're similar to runners in strawberries. If you've ever seen how strawberries can reproduce asexually, they send out a runner, like a long stem, which will then, when it touches the substrate or the soil or the bread in this case, at a distance from the original mycelial mass, or the original bunch of hyphae, then they'll be able to throw out another bunch of rhizoids, like this is over here. This is a secondary Rhizopus that's growing and starting to fruit as well. So these are horizontal, these stolons are horizontal hyphae, and they spread out over the bread surface, usually on top of the bread surface. And then these, like I said, the rhizoids then will grow downwards, and they penetrate the bread, and they anchor the mycelium, they anchor the fungus in place. Now, that's the third kind of specialized hypha then, are these sporangiophores. I'll just put that word in as well. So these things here are called sporangiophores. It's not an easy word to get your head around, to get your spelling right for. Sporangiophore. Sporangiophores are specialized hyphal stalks, and they're formed for asexual reproduction. At the end of the sporangiophores, then, when they grow sufficiently big enough, you get these swellings, these capsules forming called sporangia. So I'll just put that in as well. So these are called sporangia. It's a sporangium. And like I said, they're capsules that will be spores will be made by mitosis, all asexually produced. So mitosis will form these haploid spores. What I didn't mention, and it'll become more important later on when we look at sexual reproduction in Rhizopus. All these hyphae, this whole organism, and this is the true for Rhizopus for most of its life, most of its life cycle, and most of its most examples or whatever, it'll spend its life as a haploid organism. So all of these hyphae you see are haploid. The sporangia then will produce, by mitosis, they'll produce haploid spores. So these haploid spores eventually will get released, and the release is aided by a couple of things. First of all, there's a plate at the top here. So I'll just put in a little label here. At the very top of these swellings, you get this. You get this plate called a columella. Sorry. Just put the pen back on. It's a columella. C-O-L-U-M-E-L-L-A. Columella. And this is a plate that the sporangium forms on. And underneath that plate, you'll have this swelling. And I'll just put a little thing here. This is called an apophysis. Apophysis. All difficult words. The apophysis is a swelling underneath, and it helps with the release of the spores by getting bigger and bigger, and I suppose the pressure and the swelling gets big enough, then it causes the rupture, causes the bursting of the sporangium, of this capsule here. And this is one here that's burst, where you can see the apophysis has kind of deflated. It built up as a swelling, and then now it's just flattened. It's deflated like a deflated tire. And the reason for that is because it's burst along with the sporangium, the capsule that was around the spores, and now the spores are released. So you could call this is like self-dispersal, as you'd see it in plants with plant seeds. So these spores then will land and they'll germinate by mitosis, and you'll get more haploid hyphae growing out of them, and on it goes. So this is the asexual reproductive life cycle of Rhizopus. Okay, so like I said, most fungi, including Rhizopus, they'll stick to this method for most of their life, most of the time. If conditions get adverse, if things get difficult, then they'll switch to they'll switch to generally, and they'll generally wait until things get adverse. All right, so we're looking at a diagram here of the life cycle of a zygomycete, something like Rhizopus, and as we said, it generally will stick to an asexual reproductive method, but here's how it will reproduce asexually. So these are two, and they're coloured pink and green. These are two different compatible mating strains. They're both haploid, and they're both very similar to each other, but they don't have male and female fungi. What we do talk about is compatible mating types, and they're referred to as plus and minus. You can see here this is a mating strain minus, and this one here is a mating strain plus. It means just that they will be attracted to each other. They'll if they grow in proximity to each other. So these are just growing their everyday life up here, their everyday asexually reproductive life. They're producing sporangia, all by mitosis, asexually. Like we looked at, they produce spores and everything else. If two hyphae of two compatible mating strains grow close together, then what will happen is that, as you can see here, they will form a kind of bridge. They will touch each other, and then they will form these swellings. And these swellings are called progammetangia. I'm just going to put a little label in there that isn't on this diagram. These are called progammetangia. Each of these then constitutes a progammetangium. So they are progammetangia, plural. So you just stick that in. These are called progammetangia. And you can see that they're starting to swell. They will eventually form these cross walls which will separate them, septae, these cross walls that will separate these from the rest of the hyphae. Before they do that, nuclei, and remember the nuclei aren't separated into different cells in these hyphae. The nuclei will move in here, and that's what causes the swelling. So they become multinucleate progammetangia, like I said. And they're held on these little sort of supports here. These supports are called suspensor arms. So I'll just put that in as another little label. Got these things called suspensor arms. Okay. Suspensors. Okay. So another one there as well. Suspensor arms. There's another one just there. Okay. So you've got these swellings called progammetangia. As they develop then, like I said, you get these cross walls which separate these gammetangia, they'll now be called. They separate the gammetangia from the rest of the hyphae. So once these cross walls have formed, they now go from being called progametotangia to being gametangia. Gametangia are basically the equivalent of gametes in other organisms. They'd be multinucleate though. So what happens then as these two things develop, you get this fusion event. And so fusion of gametangia, fertilization, in other words, of gametes, you get fertilization, fusion of gametes, you get a zygospore, which is the equivalent of it is a zygote, just like fertilization in plants and in other and humans and other organisms. In this case, it's called a zygospore or a zygosporangium. A zygospore is what it's generally called. A zygospore then is a very resistant structure when it's mature, and so this is a diploid structure now. So we have this, and you can see here this section here is this slice of the reproductive cycle, going from here, or sorry, starting here as they fuse. You get a fusion event. It's a 2N structure. It's a diploid structure until meiosis occurs. So this zygospore is short-lived. At least short-lived in the sense of it's only a very small part of the reproductive cycle of the Rhizopus. It is very resistant though. It can last quite a long time until conditions are suitable. Once conditions are suitable, the mature zygospore, this is a diploid structure, just to make that really clear, this is a 2N structure, and each of these is haploid. Okay, and we don't care what the number is. In humans it's 23, this would be 2N would be 46. So we spend most of our life in this diploid structure, and you can see that fungi, in this case Rhizopus, spends its life largely in a haploid mode. And so the 2N, the diploid structure, the zygospore, is only a very small part of its life cycle. It will germinate then by meiosis. So during germination you get this meiosis event occurring. Remember, in humans meiosis is used to generate gametes. So here it's used in a completely different way. It's used here to germinate this zygospore. So this zygospore is full of diploid nuclei, and they will divide by meiosis to produce these hyphae, and the hyphae will grow and produce sporangia, and we're back to our old story then of the producing spores, which are haploid, and they will germinate and produce haploid hyphae.